I first want to say thank you. I'm very happy to be a part of this session today and share some of my work with you. As we all know, global temperatures are dramatically rising. Simultaneously, the frequency and intensity of heat waves is increasing as well. This past summer has been brutal for many of us, and wild animals must cope with these short-term bursts of heat too. But questions remain as to the mechanisms of thermal tolerance and as to which individuals will be most affected by heat. Setting aside heat for a second, we often think of body size as positively correlated with performance. For example, larger size has benefits for obtaining territories, attracting mates, or dealing with other challenging conditions. At the same time, heat may put larger individuals at a disadvantage because larger bodies retain heat more and are harder to cool. This effect was recently observed in thick-billed murres. Choi et al. Found evaporative cooling efficiency decreases with increasing body mass, suggesting that larger murres are more vulnerable to heat stress. So climate change really has the potential to change how selection acts on body size. I tested the hypothesis that larger animals are more impacted by heat in tree swallows, specifically focusing on nestlings, as they are undergoing rapid exponential growth and development. In this experiment, I simulated a heat wave inside nest boxes to look for effects on chicks' behavior, growth, and physiology. I increased the temperature of the nest by at least 2 degrees Celsius with air-activated warmers during the peak of growth starting on day 6. I recorded and measured chick behavior on that first day, day 6, and then 3 days into my experimental heat wave on day 9 post-hatch. I took blood samples and morphological measures on days 6 and 12, which also allowed me to sample across amounts of exposure to heat from a few hours to 6 days. I'm going to walk you through the behavior data first, and then I'll shift to the physiological data, looking at main effects of body mass, and then the degree to which treatment effects were mass dependent. I first measured chick thermoregulatory behavior, specifically time spent panting, and you can see a chick panting in the bottom corner of this video. I found that initially after just 1 hour of heat, panting increased in heated birds. However, this effect goes away after 3 days of heat exposure, suggesting acclimation. I also observed a similar pattern of acclimation in chick huddling. Heat also had an effect on mass. There was a main effect of treatment on chick mass in which heated birds were larger than controls. So how are these large chicks responding to heat physiologically? The first physiological response to heat that I focus on is heat shock protein or HSP gene expression. HSPs are known to prevent cellular damage and promote recovery from heat. The literature largely considers higher HSP abundance after heat stress as a protective response to incurred cellular damage. Therefore, I predicted that heat would increase HSP gene expression and that this effect would be strongest in larger chicks. The second physiological mechanism I measured is telomere length. Telomeres are the protective ends of chromosomes which shorten due to stress, aging, and accelerated growth. So I expected that heat would shorten telomeres, perhaps especially so in birds that grew the most during this heat wave. Using qPCR, I quantified relative telomere length and relative gene expression of HSP90B1, an HSP that has been linked to hyperthermia in birds. As you'll see in the upcoming results slide, however, I didn't find an overall main effect of heat on HSP gene expression, so I'm going to focus the remainder of the talk on potential mass treatment interactions. I found that in day 6 controls, there was a negative relationship between mass and HSP90B1 gene expression. So if heat is impacting largest chicks the most, we might expect this negative relationship to flatten or even reverse in heated birds. But in fact, after 4 hours of heat, I found the same relationship between HSP gene expression and mass in heated nests. larger chicks tend to have lower expression of this heat protective mechanism, which didn't respond acutely to this mild heat wave. Now remember, this is really early on, after just a few hours of heat exposure. What happens after 6 days of heat? In day 12 controls, there was no relationship between HSP90B1 and chick mass. However, the heated birds at this point have experienced 6 days of heat. So if heat effects accumulate more in larger chicks, I would expect that HSP abundance would be higher in larger experimental chicks. And this is pretty much what I found. Mass marginally predicts HSP expression with larger heated chicks having higher HSP abundance. When controlling for the random effect of nest, this result is not significant, but it is close. Lastly, when looking at telomere length on day 12 at the end of the experiment, I observed no relationship between relative telomere length and mass gain. I saw the same null result when looking at just mass as well. So in spite of the fact that the heat wave had an effect on mass, there was no relationship with telomere dynamics. Taken together, these results suggest that the heat wave initiated thermoregulatory behaviors which may have buffered chicks from mounting an initial physiological response. However, as heat continues, large chicks may be more impacted, as I saw a stronger HSP response in larger chicks on the 6th day of heat exposure. Further investigation is needed, though, to tease apart questions of exposure versus ontogeny. Because this experiment was designed to be pretty mild, in the future I will be looking at these same patterns in response to a more extreme heat wave that may push animals towards their limits of behavioral and physiological plasticity. With that, I would like to thank my lab, funding sources, and all of you for listening.